My name is Jessamyn Fairfield, and I work in the Bolin group at CRANN with metallic nanowire networks. So we look at nanowire networks to try to make novel electronic devices. There are a lot of potential applications for our work, like new types of electronics, new forms of computer memory, or also electronic circuits that function in a way similar to how the brain functions. There are a lot of groups around the world that work on devices made from individual nanowires, but we're one of the only ones working on films of nanowires altogether, because at CRANN we've got a special facility for spraying down these nanowires, so you can make like transparent conductors that way from films of nanowires. And the electrical properties are really interesting because you've basically got junctions in between the nanowires that are controlling the electrical behavior. So we make devices from those by putting down electrical contacts and then putting an electric field across the film of nanowires. The materials that we use are a variety of metallic nanowires: silver, nickel, copper, and they tend to be like tens of nanometers in diameter and then microns or a few microns in length. What we find magical about these materials is that we're putting a whole bunch of nanowires together, but the properties of the film made up of all these different nanowires aren't just the sum of the properties of individual nanowires. So basically the junctions between the nanowires are really what determine the properties of the film, and there's a whole distribution of properties for those junctions, so that basically when we send charge through, the electrical behavior of that film is determined by what's happened in the past, so it has a sort of memristor type effect. One of the major applications is trying to find new paradigms to make electronics. So different forms of both circuits for computing and memory. But the application that we're the most interested in is this sort of neuromorphic computing, like trying to make a computational circuit that's similar to the way that the brain works. I mean, there's first just the intellectual interest of trying to build computational circuits that are closer to the brain and try to understand both more about the brain and more about different ways that we can make electronics. But there are also some computational tasks that computers are much worse at than people. Like one of these is facial recognition. And so one of the hopes is that if we can build a circuit that's more capable of learning, that we can teach it to do things that tasks where humans still outperform computers. So far, we've been focusing on proof of principle type things, devices that just use the nanowire networks in a very basic way to make sure that what we're trying to do is feasible. But the next step is to really go up in terms of complexity and try to make a circuit that really has some interesting properties and that can maybe be taught in more than just the basic way that we've done so far, like try to do something that's really optimized for a specific task, or that shows more that we can go up to the complexity, of an actual brain rather than just a two-terminal device on a sheet.
Hi, my name is Arlene O'Neill and I'm a PhD researcher here in Professor Coleman's group, who is a principal investigator both in CRANN and the School of Physics. I am a nanomaterial scientist, which means that I work at the nanoscale, which is 100,000 times smaller than one single strand of hair. And I go down to that scale every day because the materials, once we go down there, behave far superior than anything we have in our world. So I take something like a graphite crystal, which is the center of our pencil, and I break it from something from our world down to its nano-sized component. And when we do that, we have a new material called graphene, and graphene is magical. It itself conducts electricity much faster than anything. It's also the strongest material ever discovered. It's completely invisible. It's stretchy. It's super. It's just, I could go on and on. So I personally prepare the material. I break it down into its single sheets of graphene. But once we have it at that scale, we can mix it with polymers. And polymers are just plastics. So we can make them really, really strong by the addition of these sheets of carbon, which are the graphene sheets. There's also a field of flexible electronics. So again, mixing these nanosheets in with plastics, we can potentially run a current through a plastic such that eventually what we do hope is that they will go into the screens of computers, televisions so that they're going to be flexible. We're going to be able to roll them up, put them in our pocket and take them back out. Graphene is great, and it was the very first two-dimensional material, but really what it sparked was that we could perhaps apply this to other layered materials, and we do that to a group of materials called transition metal dichalcogenides. And what that really means is, although graphene is great, we are limited by the fact that it's one material with finite properties. But when we extend the theory of making two-dimensional materials to these transition metal dichalcogenides, we have at least 60 new materials that each have their own unique exotic properties. For example, in thermoelectrics, where they will convert heat back into electricity extremely efficiently. Some of them will store energy really, really well. So the next generation of batteries, we all know we need to take our iPhone, charge it up every night. Potentially with these materials, we're going to have batteries that will last for months. The group that I work in is Professor Coleman's group, and his research has been really highly recognised around the world lately. We've got some great publications that people are citing from everywhere in the world, and it's great to be Irish, a small little island where we're recognised internationally for this. Plus, it's also like a little mystery when you go into the lab and you test these things for the first time. No one has ever done it before. So it's great to be involved in an environment that's so cutting edge, that's a really pushing the frontiers of science.
My name's Jensen. I'm working in the field of nanobiophysics. I'm a PhD student in the research group of Professor Martin Hegner in CRANN, Trinity College Dublin. I'm associated with School of Physics. I work with silicon cantilever arrays to do nanobiodiagnostics for medical applications. I work with silicon cantilever arrays. They're like small diving boards that are sensitive to a mass landing on the surface, and I use them in conjunction with nanomaterials such as small gold spheres or magnetic spheres to do diagnostic tests. I work with small silicon cantilever arrays. They're like small diving boards, and we shake them up and down, and when something lands on the surface, the properties of the silicon change, which allow us to detect how much has landed on the surface. And then I also use small nanoparticles in the order of 50 nanometers in diameter to put them into biological solutions such as a blood sample, and then the properties of the coating on the nanoparticle allow me to detect different materials from the blood, for example a bacteria or a protein, and then when the nanoparticle lands on the cantilever I'm able to detect it and say if you have a disease or not. So these silicon cantilever arrays that I work with, they're exceptionally small. They're 80 times thinner than a human hair, and they're only about 0.5 millimeter in length. The small size means that they're very sensitive for the different tests that I'd perform, and they're able to pick up very, very small amounts of mass. For example, the sensitivity is around 1 nanogram, which is a billionth of a gram. What makes our cantilevers exceptional is that we can change the surface of the cantilever to suit different tests. For example, we can test for the presence of protein from Parkinson's disease. We can grow different bacteria and fungi on the surface, like for an agar plate. And we can also do genomics testing for different RNA strands and DNA strands. What makes the work that we do exceptional is that we work in a complete liquid environment. So we shake the cantilevers up and down in liquid, which is very difficult. It adds its own set of problems compared to how other groups in the world would do it. But on the other hand, it does provide a more clinically relevant test. So you can imagine that biology doesn't work in a vacuum. So if you want to test for a biological molecule, you'll have to do the test in liquid to get the right results. A conventional test would involve a large blood sample. So like if you go for a test in the doctor, you'll get tens of milliliters extracted from your body. We'd hope to do the same sort of tests with a few drops of blood, basically. So the main aim of the group is eventually to commercialize this technology to make easier tests for example, for a GP or bedside testing in hospitals. Or also some of our applications involve in speeding up tests which will be relevant to the pharma industry.
My name is Patrick Cronin and I'm a postgrad researcher in the Materials and Surface Science Institute at the University of Limerick. The purpose of my work is to develop a material or textile technology that will instill antibacterial properties into such textiles. So internationally, this is a very novel technique. It is a binderless method, it's very cost effective, and it uses inexpensive processes. We coat our textiles with titanium dioxide nanoparticles, and what's magical about these nanoparticles is their photocatalytic abilities and therefore their abilities to decompose bacteria and eliminate it from the textile surface. The benefit of this work relates to incorporating these textiles into the medical environment. Currently, 1 in 14 people will contract a healthcare associated infection within the Irish healthcare industry. These textiles aim to lower that percentage and reduce the quantity of bacteria in hospital environments. Primarily, bacterial infections occur on personal protection equipment of medical practitioners. The main areas this occur are around the pockets, around the sleeves, where the most hand contact occurs. Currently, we've developed a technology towards targeting bacteria such as MRSA and E. coli within healthcare environments. The future of these textiles is very broad. We can target public areas such as creches, trains, wherever people come in contact with surfaces that multiple people have been in contact with. The goal of the research at the moment is to broaden the horizon of the bacteria that we can eliminate. The nanoparticles we use are a photocatalytic material, therefore they use a certain wavelength of light and they require a certain wavelength of light to eliminate the bacteria. If we are aiming to broaden the horizon, incorporating nanoparticles that require different levels of light and also different levels of methods of killing bacteria. The area of my work is incredibly exciting because I get to work with materials that are so far advanced. They incorporate technology that is incredibly novel and materials that in 20, 30 years' time can really change the landscape and change the way that we treat our environment.